Hello, everyone. It's a great pleasure to introduce our speaker today, Dr. Roger Greenberg from UPenn. Many of you may have known Roger from his time at Dana Farber at Harvard Medical School. Roger did his MD, PhD training at Albert Einstein. He did his PhD with Ron DePino. And then he came to Boston and he did his residence at Brigham's and his postdoc training with Dr. David Livingston at Dana Farber. So in 2007, Roger joined the faculty of Penn and quickly rose through the ranks, where he's the full professor now. He's also the director of Penn Center for Genome Integrity. He's also the director of basic science at the Basic Cancer for BRCA. And he's also a co-leader of the breast cancer program at the Abraham Cancer Center at Penn. Roger's lab has done many important studies on the DNA repair pathways. In particular, Roger's lab has studied BRCA1 and BRCA2 and helped to understand how these tumor suppressors regulate double-strand break repair. And Roger's lab has also studied non-canonical BRCA-independent DNA repair pathways, particularly the alternative lengthening of telomere pathway. Alt pathway. So Roger has designed actually several very creative assays to follow DNA repair at telomeres. And some of you may know Roger's recent work that linked DNA repair, DNA damage to micronuclei and activation of cGAS pathway and immune response. That's again a very important finding in the DNA repair field. I think today Roger will tell us another important area his lab is working on, and that is the regulation of chromatin structure by DNA damage. He will tell us about the new story, a new type of chromatin-related vulnerability of cancer cells that can actually confer PARP inhibitor sensitivity. Okay, so before I give the stage to Roger, I just want to remind you that please visit this website of Connect Science so you can find a list of future speakers, and also you can find the recorded talks from the previous speakers. And I also want to remind the audience that, so if you have questions for Roger, please type it in the Q&A box, not the check box. I will read out your questions at the end of the talk. So without further ado, Roger, please feel free to start.
All right, Lee, thank you very much for the invitation. I would say it's always good to be back at Dana Farber and see all of my old friends, and I guess this is about the best we can do for now. But Lee has certainly become a good friend, and I think our interests really overlap and complement each other quite a bit. And I'm definitely an admirer of all of his accomplishments. So today I'd like to tell work that was recently published. I think it's probably the most appropriate for this series. I apologize for those who read it last month, but you'll have a chance to see it again. And it's really the creation of an outstanding postdoc in my lab, Priyanka Verma, who is now in the job market. And I really can't say enough good things about her. And in collaboration with a colleague of mine in the Department of Cancer Biology at Penn, Jinwei Shi, who's pioneered a number of CRISPR screening methods. And I'd also like to highlight our beautiful campus, including Ben Franklin, and this sculpture of BRCA2 performing homologous recombination entitled Homologous Hope, hanging from the Perlman Center. So our genomes accumulate numerous DNA lesions with every single cell cycle. And this presents a particular challenge in S phase when DNA replication encounters them. For example, there are, thought to be up to about 10 to the 6th incorporations of ribonucleotides per every cell cycle. There are damaged bases, abasic sites, single-stranded breaks, more than 10,000 per day. And there are even more lethal lesions such as interstrand crosslinks and double-stranded breaks. When replication forks encounter these, this creates a number of problems that they have to deal with, including uncoupling of the replicative helicase and polymerases, as well as compensatory type of responses in which these forks can regress back into this interesting chicken foot structure shown from a nice review from Massimo Lopez and Alessandro Vendini. One of the most important ways of dealing with this type of lesion is through an evolutionarily conserved DNA repair reaction in S phase known as homologous recombination. Here I depict a replication fork encountering a single-stranded break, and this would lead in a BRCA1 and BRCA2 dependent manner, seeding RAD51 onto this single-stranded DNA element to create a nucleofilament that can perform a homology search within the duplex DNA, typically of the sister chromatid, capture homology, and copy along in a templated manner, or a copy and paste type of mechanism for what is in principle an error-free mechanism of DNA repair. Now, human genetics tell us the importance of this, not only for genome stability, but also for tumor suppression. Mutation of BRCA1 or BRCA2 are the most common causes of hereditary breast and ovarian cancer. When I was a postdoc with David Livingston, we showed that BRCA1 was in multiple protein complexes that we dubbed the BRCA tumor suppressor network. And it turns out that genes encoding many of their interacting partners are also mutated, both in the germline and somatically. In a number of cancers, and in particular in high-grade serous ovarian cancer, which is thought to be about 50% of it is thought to be homologous recombination deficient. Now, this not only relates to cancer etiology, to the etiology of several cancers, but also presents a new vulnerability that can be exploited according to the concept of synthetic lethality. This is based on really a simple idea of genetic buffering. And in relation to this, non-essential genes, for example, gene A or gene B, when they're mutated and not functional, can still permit viability of a cell or an organism, but the combination of these two together, loss of both gene A and gene B, would result in lethality. This was observed in genetic experiments in Drosophila almost 100 years ago, and was quickly, or later on, I should say, realized that this could be applied for cancer therapy by Lee Hartwell and several others. And so in real terms, this applies to BRCA1 and BRCA2 mutant cancers, as well as homologous recombination cancers, deficient cancers more generally, as first shown by landmark studies from both the Ashworth and Halliday groups. The typical BRCA carrier is heterozygous. Every normal somatic cell in these individuals can perform homologous recombination pretty well. On the other hand, the tumor generally has allele-specific loss of heterozygosity event where it will lose the wild-type allele, creating an effective BRCA null state in the tumor, whereas the non-transformed cells still have this intact. And this creates about 100-fold or more increased sensitivity to poly-ADP ribose polymerase inhibitors in the null state in comparison to the heterozygous. Now, what we can see here from the SOLO-1 trial is data in high-grade serous ovarian cancer that had originally responded to platinum and then treated during the maintenance phase with either these patients with either the first approved PARP inhibitor, olaparib, or a placebo control. And what you can see here is about a 36-month increase in progression-free survival in the olaparib-treated patients. There are now 4 FDA-approved PARP inhibitors, and they work. Really, it seems to be irrespective of tissue type in homologous recombination deficient ovarian, breast, prostate, and pancreatic cancers. But there are problems with this as well, and that is that about 50% of patients don't respond de novo, and almost all of those that initially responded will eventually acquire resistance. And so there are need for new agents that can overcome these resistance mechanisms, and I'll talk about some of our data that relates to this problem. But before I do, I'd like to get to the question of how PARP inhibitors really could work. Poly-ADP ribose polymerases, there are a large family of them in the cell. But the PARP1 and 2 enzymes are the ones that are most intimately connected to DNA damage responses. And what they do is recognize a variety of different genomic lesions, indicated by this star here, including single-stranded breaks, double-stranded breaks, abasic sites, replication forks, or even bulky DNA adducts. When PARP binds to DNA, this turns on its enzymatic activity about 10,000-fold through large sort of structural changes to the enzyme that allow it to be active, creating polymerization of NAD into these branch structures that I indicate in orange here. This can recruit a number of different DNA repair factors that have PAR binding domains, allowing them to localize to DNA damage sites and perform a variety of different DNA repair events. Now, an interesting and less studied feature of this is the longstanding observation from Guy Poirier in almost 40 years ago now, in which polynucleosomes incubated with PARP1 and NAD would decondense into these structures, and so the PARylation has a large effect on chromatin structure through decondensation. Now, although this has been known for some time, its relationship to PARP inhibitor responses in the context of homologous recombination deficiency had really not been explored. And so the questions we had at the start of this study is, would the PARP-dependent chromatin remodeling be required for the viability of BRCA mutant cells? And if so, could it be targeted to enhance PARP inhibitor responses and overcome resistance? And obviously, the best way to do this would be to know what was responsible for this aspect of PARP activity. And finally, in relation to all of this, what would actually be the endogenous lesions that PARP has to repair in order to sustain viability in BRCA mutant cells? And so we took on this in a relatively unbiased way. We performed CRISPR screens where Priyanka learned all those CRISPR screening methodologies from Jinwei based on an approach that Jinwei actually developed when he was a graduate student with Chris Vakoch at Cold Spring Harbor. And this method, what they use, the beauty of it is that they target functional domains within proteins, and this creates a higher efficiency CRISPR screening because there's very little play for indels to actually create a functional protein when the guide RNAs are directed to functional domains. And so we directed our library to chromatin-associated factors with the idea that one or more of these would be responsible for PARP-dependent chromatin relaxation, and we could see how this could affect the PARP-dependent response. We did this with about, I think, to about 179 genes using either a vehicle control or a lethal dose 20 of olaparib. And about 14 days later, did deep sequencing to look at the guides that were either enriched or lost in these populations. We also performed these studies in BRCA1 and a panel of different BRCA1 and BRCA2 mutant cells. I'm only showing two of them right here. And importantly, the lethal dose 20 of olaparib allows us to find hits that will increase the sensitivity to PARP inhibitors. To summarize our findings, the best hit in this screen is a SNP2-like chromatin remodeling ATPase known as ALC1, and this was generally the best hit across every single cell line that we looked at in conveying PARP inhibitor increasing PARP inhibitor sensitivity. And we performed the validation assays using a convenient approach in which we have GFP co-expressed from the Cas9 and the guide RNA vector. And what that allows us to do is look for loss of GFP over time to establish either synthetic lethal or synthetic sick or PARP inhibitor sensitivity type of relationships. So just to show you our data, we took a panel of isogenic BRCA1 or BRCA2 mutant lines, plus and minus each of the alleles here. In these RP1 cells that we got from Dan Durocher's lab, what you can see here, the color scheme is consistent throughout. In light blue is ALC1, loss of ALC1 alone, and in dark blue is ALC1 plus PARP inhibitor. whereas in dark gray, we have PARP inhibitor alone. And what you can see is that there's a synthetic sick relationship between ALC1 and either BRC1 or BRC2. And I describe this as synthetic sick because we can still get viable cells. I don't really think it's a strong synthetic lethal interaction, but the cells do have a slower growth over time. But we see a profound PARP inhibitor sensitivity when we combine this with even very low doses of PARP inhibitor at 1 nanomolar of olaparib that won't even really touch these BRCA1 or BRCA2 null cells by themselves if ALC1 is intact. Now, also notably, we see a very small or very modest effect when homologous recombination is proficient in the BRCA wild type cells seen here and here, and that's consistent across lines. Now, we did a further characterization using a dual Cas9 system to study epistasis with other DNA repair pathways, and these are actually clean systems. It's not double knockdown, which is notably fraught with problems when trying to do epistasis. We have a complete knockout of ALC1, and then we knock out the other repair pathway in question here. And what you can see is a small synthetic sick relationship with other factors involved in replication-associated homologous recombination and a profound PARP inhibitor sensitivity. Interestingly, we see a synthetic sick and PARP inhibitor sensitivity with the single-strand break repair factor XRCC1, which will relate to mechanism that I'll get into later. But we don't see any effect when we combine this with, for example, loss of non-homologous end joining or nucleotide excision repair, really other base excision repair factors as well. And so ALC1 seems to specifically relate to factors involved in homologous recombination. But it is not generally required for homologous recombination repair. And so I bring up this data right here because clinically, response to platinum is the best predictor of response to PARP inhibitors in BRCA mutant cancers. And yet we see, I think as this audience well knows, platinum elicits BRCA-dependent homologous recombination. And what we can see as opposed to PARP inhibitor, where loss of ALC1 creates a large window, we don't really see any of this. There is no impact irrespective of BRCA status, either wild type or knockout, in relation to response to platinum or in fact other agents such as camptothecin as well that require homologous recombination. And what that told us up front was that ALC1 is responding specifically to events that might involve PARylation. And that made a lot of sense to us based on the structure of ALC1. I should point out first that ALC1, which is also known as CHD1L, it stands for amplified in liver cancer, was first described by Simon Bolton's group as well as by the Conaways. And they had very similar results in both of their early studies. What you can see is at the N-terminus, ALC1 is a SNF2-like ATPase activity that has bona fide in vitro nucleosome sliding that depends on its ATPase activity, where it can move nucleosomes along on DNA. And what is unique about it in comparison to the rest of this class of enzymes is that it has a C-terminal macrodomain that binds very tightly to poly-ADP ribose chains, and shown by several, including the Lederner group, showed that this macrodomain binds to tri-ADP ribose with about 10 nanomolar KD. And so this also has an important function. The macrodomain targets it to sites of DNA damage, and you can see this nicely by this laser stripe experiment done here by the Bolton lab, in which you can see wild-type ALC1 rapidly getting to these laser-induced stripes of single and double-strand breaks, and it starts to diminish quickly after that, mirroring the kinetics of PARylation and deparylation that would look very similar to this. The ATPase activity doesn't affect its recruitment. In fact, it lingers longer, perhaps because of a lack of DNA repair going on, whereas mutations within the macrodomain abolish this localization to double-strand breaks. The macrodomain also has other important functions, including that it is involved in stimulating the nucleosome sliding activity of ALC1, If you incubate this with PARP, what it does is it removes an autoinhibitory conformation here, and also it allows ALC1 to recognize these nucleosomes and slide them more efficiently. All right, so we then looked at both of these sort of modalities within ALC1 and what was their relationship to this PARP resistance phenotype. Here we created several mutants, either this K77R ATPase deficient mutant. We also created mutants within this region right here, which interacts with the histone H4 tail and is required for nucleosome sliding in vitro as well. Now, I'll just show the ATPase activity, but the results are consistent for both. Even with very low doses of olaparib right here in the knockout of ALC1, we almost eliminate colony formation. We can restore this with wild type, but the ATPase mutant or the histone tail binding mutants do not restore resistance. And we see a similar effect when we make mutations within the macro domain. We had to make 2 missense mutants to actually get this effect, where we completely eliminate colony formation if the macro domain no longer binds PAR. If we even have residual PAR binding, we actually get colonies forming here with a single point mutant. And so that tells us that both of these are required. We then ask the question, would this increased sensitivity to PARP inhibitors have an effect in a tumor formation in an in vivo model? We used the triple negative BRCA1 mutant breast cancer line, SUM149PT, which is only modestly sensitive to PARP inhibitors in vivo. When we infect with guide RNAs to ALC1 or to a negative control, we select for about 5 days, allowing these tumors to grow, and then we treat with either a DMSO vehicle control by oral gavage or olaparib 5 days a week. And what you can see here is that olaparib in the controls only extends about 1 week before tumors start to take off. ALC1 deficiency was better than 50 mg per kilogram of olaparib, and when we combined ALC1 loss with olaparib, the tumors grew very, very slowly. And in fact, eventually they did grow out and the mice succumbed to them, but when we examined the tumors, this was because of incomplete deletion of ALC1, and we saw expression on a Western blot of ALC1, suggesting that it really was required for the formation of these tumors over an extended period of time. So this all raises the question of how do PARP inhibitors work, and how could loss of ALC1 profoundly increase sensitivity, specifically of homologous recombination deficient cells to them? And moreover, could loss of ALC1 bypass or mitigate some of the problems with a number of the known resistance mechanisms? So as I talked about before, PARP1 and 2 are the dominant enzymes in PARP species involved in the DNA damage response. And in particular, PARP1 is expressed at a much higher level and seems to be predominantly active, whereas PARP2 has a more minor role. These respond to a variety of different lesions, which activates PARylation and helps repair these lesions prior to entry into S phase. Although they work in S phase as well. If PARP1 is lost or PARP is inhibited, instead, these lesions persist and they're shunted into S phase, requiring homologous recombination dependent repair. Now, in a homologous recombination deficient state, instead, what happens is these replication forks accumulate, they can collapse into double-strand breaks, or they can perform a reaction together with other stalled forks or double-strand breaks, creating toxic non-homologous end joining as a component of the cytotoxic response. Now, another important aspect of how PARP inhibitor mechanism is that it traps PARP1 and 2 onto the DNA, as originally shown by Yves Pommier's group. What happens is PARP autoparylates, and that allows it to release. And what PARP inhibitors do by inhibiting this enzymatic activity is that the PARP enzymes bind very tightly to DNA and are maintained in this state in a PARP-trapped manner, which also contributes to toxicity. And so this model has a number of important predictions that have been borne out in the literature. Loss of toxic non-homologous end joining prevents these radial chromosomes. It also gives extra time, particularly in a BRCA1 mutant cell, in increased end resection that can allow HR to occur in a BRCA1 independent manner, as shown by Andre Nuzenzweig, Jos Jonkers, and others. Loss of PARP1 also causes resistance because of the trapping effect right here. And probably the most important mechanism in patients of resistance are reversion mutations that restore reading frame in BRCA1 and 2 and allow homologous recombination to proceed. And so we looked at this specifically based on this model. Would this sort of feed-in mechanism into toxic end joining exist in ALC1 mutant cells in the context of HR deficiency? And indeed it does. We see numerous radial chromosomes and breaks just as predicted, and loss of ALC1, as you can see in dark blue, increases the number of breaks even at baseline in the absence of PARP inhibition, but this increases even more after olaparib treatment, and particularly we see a large increase in these fusions. So the question arises, would it overcome resistance from 53BP1 shield and toxic end joining type of reactions right here? And so what you can see on the extreme curves of these are loss of ALC1 in a BRCA1 mutant ovarian cancer line where you have sensitivity compared to the parental control. Here you have loss of Rev7 alone where you get resistance. And if you combine the two with ALC1 and Rev7 together, what you get is an intermediate effect. It is relatively resistant, but still within the sensitive range. And we would predict that this would still result in clinical sensitivity based on it having very similar sensitivity curves to olaparib, as does the parental control lines. And in fact, even with reversion mutations, which we obtained a number of different BRCA1 revertant cell lines from Neil Johnson, we also still maintain somewhat of a therapeutic window when we use strong PARP trapping agents. So here, this parental line, SUM149PT, has a mutation within the exon 11 region, and what Neil did is use CRISPR on either side of this to create breaks and to generate revertants that religated this in frame to create a full-length protein. You can see ALC1 loss alone creating sensitivity here in the parental controls. Here's the revertants where you have a very large effect on resistance. And if we knock out ALC1 in this, again, it is not as sensitive and it is as ALC1 loss alone, and it is in fact not even as sensitive as the parental BRCA mutant controls, but there still is a therapeutic window right here, consistent with the idea of that increased PARP trapping per se might be able to overcome some of these problems even when HR becomes restored in cancer lines. All right, and so finally, I'll get to a more interesting observation that we had regarding PARP trapping. All right, so as I had mentioned earlier, loss of PARP1 creates resistance to PARP inhibitors, typically in BRCA mutant lines, by removing this trapping effect. There is a somewhat of a synthetic lethal relationship when PARP1 is lost in a BRCA mutant cell, but it is not as much of an effect as the pharmacologic trapping of PARP1 on chromatin. And you can see this right here in this example where we have a parental control in gray that is BRCA1 mutant. If we remove PARP1 from it, these cells become resistant to PARP inhibitors and do quite a bit better. Here is ALC1 loss alone, where you have again, a large therapeutic window until very high doses of PARP inhibitor are used. So how about the double knockout of ALC1 and PARP1? All right, interestingly, these cells maintained sensitivity to PARP inhibitors and to olaparib or talazoparib until we get to very high concentrations of about 1 micromolar of PARP inhibitor, something that we, nearly 1,000 times more than we were, 100 times more than we would need to use. All right, so that suggested to us that that ALC1 might be a little bit different. Loss of ALC1 might create different dependencies than are normally found in BRCA mutant cells in relation to the PARP enzymes. And what it suggested is that Perhaps in the absence of PARP1, PARP2 is also being trapped. Now this, as I mentioned, doesn't play much of a role in the response normally. And you can see this in these BRCA1 mutant cells alone, loss of PARP2 on top of PARP1. Here's the PARP1 in gray and the double knockout is in black. There's really a minimal effect on overall resistance to PARP inhibitors. On the other hand, with ALC1 here alone, here you can see with PARP1, it's still very sensitive to these. But if we get rid of PARP1 and 2, now this curve shifts far to the right and we have complete resistance. So the effect of ALC1 depends on both PARP1 and 2, unlike normally when PARP1 has almost the entire effect is encoded within PARP1. So we can see, I've depicted this here with wild type cells entering S phase, that with PARP inhibitors, there's a trapping effect that predominantly goes through PARP1, creating DNA damage, and loss of PARP1 causes resistance. There should be increased trapping of PARP1 based on our studies in an ALC1 null cell. But in the absence of PARP1, PARP2 trapping can compensate and still create quite a bit of toxicity in combination with low doses of PARP inhibitors, as we've observed. And so this predicts that we should be able to see this, and we did this, tested this hypothesis using immunofluorescence assays. And I'd like to thank Matthias Altmaier for his advice on doing these, where we can observe a strong increase in PARP1 trapped onto chromatin in the combination with MMS and olaparib, particularly in ALC1 null cells that we see in comparison to controls. And we also see increased trapping of PARP2 in these studies, as we would have predicted. Right. So one of the things that you may be thinking with this work is how does a PAR-dependent enzyme, how does loss of a PAR-dependent enzyme actually cause sensitivity to PARP inhibitors? How could there be such an effect? Wouldn't one predict that they'd merely be epistatic? If you're inhibiting PARylation through a PARP inhibitor, wouldn't this result in a complete loss of ALC1 activity as well? All right, so just to go back to sort of some bullet points of the findings from the field, right? This binds to PARylation and it is required to get to damage sites, and the nucleosome slotting also requires PARylation. So it raises an important question. How do PARP inhibitors really work? And are PARP inhibitor really equivalent? Is a PARP inhibition really equivalent to combined loss of PARP1 and 2? And so we thought that the best way to look at this first is through a genetic approach, in which we knocked out PARP1 or combined PARP1 and 2. And we just merely looked at localization of ALC1, which we know is PAR dependent. And you can see ALC1 localizes to chromatin irrespective of cell cycle when we treat cells with the DNA alkylating agent methylmethane sulfonate. PARP1 loss reduces ALC1 onto chromatin by about 80%, but we can still see about 20% of the levels of ALC1 in comparison to baseline in these knockouts. PARP2 had a minimal effect. It accounted for about a 20% loss when we have a PARP2 knockout of ALC1. And the combined PARP1 and 2 knockout completely eliminated ALC1 association with chromatin. And so how does that compare to PARP inhibitors, even higher doses than we would normally use? So we looked at this from several different doses. We used 100 nanomolar of olaparib, and notably, that's 100 times more than what we need to use in the context in our experiments. Here's MMS alone right here. Here's 100 nanomolar of olaparib for 24 hours. We see a very modest reduction in the overall level of ALC1 on chromatin. And even if we crank up the concentration to 5 micromolar, we can still see ALC1 on chromatin above baseline, telling us that there's PARylation. And we think that this is actually the norm rather than the exception. It's very consistent with literature from Caldecott lab, from the Altmaier lab, and from others. in that 10 to 20 micromolar of PARP inhibitors, well beyond what is used in any of these preclinical models, is needed to recapitulate a complete double knockout of PARP1 and 2. And so what this suggests to us then at the doses that we're using is that very little PARP is actually being inhibited, all right? So let's just look at this. Here are the PARP1 and 2 enzymes. The catalytic domains are at the C terminus. Their in vitro IC50s for PARP1 is about 5 nanomolar, for PARP2, 1 nanomolar. Now, the effective concentration typically in BRCA mutant cells is higher than that. And yet we're seeing efficacy at 0.1 nanomolar in BRCA2 mutant cells. And in fact, by clonogenic assays, this is nearly 1,000 times more than what's needed in the BRCA2 mutant controls. And we see really very minimal effects if BRCA2 is wild type. And so what I think this tells us, at 0.1 nanomolar, there's probably an undetectable effect on PARylation at damage sites, and instead, a small subset of PARP1 and 2 are being inhibited and trapped onto DNA. And that is what is accounting for the toxicity in this. And what it tells us then is that ALC1 deficiency is creating the type of genomic lesions that very tightly trap PARP1 and 2 onto the chromatin as a mechanism of effective killing. And it makes us rethink, in combination with a host of work from many other labs, of how these drugs are working, and that they clearly are not working by a purely phenocopy of genetic synthetic lethal interactions. Right. So this brings us to the question of what subset of PAR activating lesions is ALC1 actually necessary to repair. Right. So it had been implicated from a number of different groups to be involved in response to base excision repair type of mechanisms that can activate PAR. And so we tested several different agents which can, both of them elicit PAR type of DNA damage responses, either the DNA base alkylator MMS or camptothecin, which traps topoisomerase 1 onto the ends of DNA. All right, now these differ notably in several different ways, including a series of base processing events to get to an abasic site, eventually nuclease incisions, which can lead to a 3' hydroxyl 5' phosphate, allowing this repair chemistry to ensue. And what we thought is that a chromatin remodeler should provide access to repair factors to more of these upstream lesions, as opposed to others that are trapping further down DNA adducts onto single-stranded DNA. And so that predicts a few things. One is that loss of ALC1 should create sensitivity probably to the MMS and not CPT. And moreover, in the absence of ALC1, this would channel this type of DNA damage into a BRCA1 and BRCA2 dependent manner, resulting in non-epistasis. And this is exactly what we observe. Loss of ALC1 alone creates sensitivity to MMS, as does loss of BRCA2, and the combination is even more sensitive, telling us that these pathways are not completely linear and that existing damage feeds into S phase. On the other hand, with camptothecin, we see absolutely no sensitivity in our assays of loss of ALC1, and there is no exacerbation of the known relationship sensitivity of BRCA2 null cells to CPT. All right. Moreover, if we have upstream lesions right here that are unresolved, it would predict that it would be different than merely double replication forks running into single-stranded breaks, but that they might encounter base lesions that they would have to skip over. All right, and that, I think, brings up how do these type of interactions result in the type of lesions that can trap PARP1 and 2 onto DNA. And I think a conceptual framework from this comes from classic work from Dean Rupp, in which discontinuities in DNA synthesis were observed when bulky base adducts were formed in E. coli, with the idea that replication forks would run into these and have to prime downstream of them, creating large single-stranded gaps. And these type of lesions have also been observed in vertebrate systems, as noted by elegant work from the Costanzo lab, as well as from Alessandro Vendini, Carlene Simprich, and others. And so we posit that these type of lesions forming during S phase when unrepaired base lesions arise would be responsible for the increased PARP trapping. And so that predicts that we would be able to see a number of single-stranded gaps in the DNA when they replicate, and this would be exacerbated in an ALC1 null cell, particularly when it's treated with PARP inhibitors. So in dark blue, we have all ALC1 null ALC1 deletion in BRCA mutant cells. In gray are the controls. And what you can see is that the fibers are shorter at baseline. All right. So just to take you through this assay, we label with 2 halogenated nucleosides. This creates DNA fibers that, when be stretched out on silanized slides, are then cleaved with an S1 nuclease. And if there are single-stranded gaps in here, this will be able to cleave these, creating a shorter DNA fiber. And what you can observe here is that uniformly ALC1 deficiency results in more gaps in the DNA, and this is really particularly evident when we combine this with PARP inhibition. All right, so I'll raise the question again, how does PARP-dependent nucleosome sliding, all right, how does PARP-dependent nucleosome sliding prevent the genesis of these lesions? So we turn to a more genomic approach to look at this in a genome-wide scale using the method called ATAC-seq. And what this uses is a transposase that can put linker DNA into accessible regions of the genome that are nucleosome poor. This is typically excluded from more dense nucleosome-rich regions. The DNA can be sheared up, it can be amplified and then sequenced. All right, and we did this according to the scheme in BRCA1 or BRCA2 null cells, where 24 hours after knocking out ALC1, we would treat with either the PARP inhibitor olaparib or talazoparib for 4 hours, and then perform the ATAC-seq studies. And so looking at this, treatment with PARP inhibitor alone results in about a 1.3-fold decrease in overall chromatin accessibility. And if we combine this with ALC1, this went down to about 3.5-fold reduction, telling us that they can cooperate to prevent accessible regions of the genome. Now, notably, these did not occur in recurrent regions. They were not transcription associated. There were not changes in the RNA-seq profile during these experiments, but rather they could occur stochastically and require metagene analysis, as would be predicted for DNA damaging type of reactions. Now, this reduction in chromatin accessibility prevents access of base excision and single-strand break repair factors from getting to chromatin. And we can see this even with loss of ALC1, we reduce all of these factors from getting there. And if we combine this with PARP inhibitor, they're reduced, their chromatin association is reduced even further. And so we have a model from this that base lesions are typically recognized very rapidly by PARP1 and 2, which can correlate the chromatin, allowing recruitment of ALC1 to slide nucleosomes and create large accessible regions that can be repaired, preventing the need for these to be shunted into S phase for recombination repair from BRCA1 and 2. Now, in the absence of ALC1 and together with PARP inhibition, what we see is a really strong condensation or failure to decondense DNA damage sites. And this excludes repair factors from getting there, increasing the burden on homologous recombination during DNA replication to repair these lesions. These results, skipping over these base lesions, results in further single-stranded gaps in the DNA and increased PARP trapping and creating more of a requirement for homologous recombination. Our work was published last month in Nature Cell Biology, and I'd also like to point out a number of studies which were quite consistent and complementary from the Bolton, Lederner, and Tominski labs. And so finally, it brings up another interesting point, which I'll end on. Does chromatin remodeling, together with PARP inhibitor, we see this cooperativity, could this expand the spectrum of agents, which damaging agents, which we could induce a response? All right. And so we looked at this in several different lines. I'll just show you one of them here, where we take BRCA null cells and we knock out ALC1 versus a control, and we treat with a series of different, from low to moderate doses of olaparib, and treat with ionizing radiation. Now, ALC1 null cells are not sensitive to olaparib. There is some discrepancy in the literature from before, but we've looked at multiple different BRCA lines. We've looked at U2S cells. We do not see sensitivity, at least in any of the lines we've looked at. In fact, if anything, they're a little bit resistant in comparison to controls. Now, on the other hand, when we combine this with low doses of PARP inhibitor, they become very sensitive. And we actually can see a synergistic interaction between low-dose PARP inhibitor and loss of ALC1. Now PARP inhibitors were originally developed as radiosensitizing agents, but we can accentuate this quite a bit by removing the chromatin remodeling aspect of this in combination with low doses of PARP inhibitors. So bringing back to the model, I think I've gone over this several times right now. Chromatin remodeling and chromatin accessibility is an essential component of the PARP inhibitor response, and it can be exploited to accentuate this in a way that can overcome a number of different resistance mechanisms, specifically in the context of homologous recombination deficiency. And it raises additional questions that we're quite interested in, including what are the endogenous adducts that are causing genomic instability in BRCA mutant cells? Could these, in fact, even explain some of the tissue specificity that they would be more prevalent in the tissues that are higher risk for malignancy in BRCA carriers? And finally, how do these DNA adducts impact cancer genome evolution? So I'd like to once again thank the people involved. This is Priyanka, who is just an extraordinary postdoc. Those of you in the DNA repair field may know that she is responsible for a number of other studies from my lab, including the alt telomere recombination that Lee mentioned earlier. And she had help from a lot of important people, in particular Jinwei Shi, who's really just a phenomenal collaborator, Babak Faryabi and Bob Mack at Penn. We are, of course, grateful for the generosity of a number of colleagues in the field who've given us important reagents, as well as for our funding, and a shout out for the Penn Center for Genome Integrity at Penn that I direct, and that I think will give opportunity to interact with a lot of others in the field. So thank you once again, and I'm happy to take questions.
Thank you, Roger, for the wonderful talk. So there are a few questions in the Q&A box, but maybe I can start with one question. So your result, S1 nuclease assay, show beautifully that ALC1 matters for PARP trapping and gap formation on nascent DNA. Do you think the function of ALC1 is specific to replication-associated gaps, or any gaps generated by BER, for example? Yes.
All right, it's a really good question, Lee. And all right, so what we see at baseline, in unperturbed cells, ALC1 is on chromatin in S phase. All right, you don't see it on chromatin in G1 in unperturbed cells. All right, and David Cortez's IPON studies, ALC1 is very prominent at replication forks and increases if you create problems with an ATR inhibitor. If we treat with MMS, on the other hand, it'll go to damage DNA in any cell cycle phase. And so I think if there are base lesions, right, there are two aspects of where it could be contributing to this. And one is, in all cell cycle phases, removing these lesions prior to S phase, all right, in G1. And the other is, it seems to be important during DNA replication. So it's a little bit of a longer-winded descriptive answer to your question. We don't know where the gaps are arising, but ALC1 seems to be most active in S phase. And if there's base damage, it can be eliminating these earlier on.
Okay, great. Thank you. So let's go to the Q&A box. The first question is, are CHD1 and CHD4 in your CRISPR library? Does guide RNase CHD1 sensitize cells to PARP inhibitor? And also, CDH4.
All right.
All right. The
Good
chromatin remodeling complex. Yeah.
Good questions. I think they are in our library. I'd have to go back and check. We had we did this in a domain-focused chromatin library, and I think they should be in there. CHD4 did come out, I believe, in Simon Bolton's screen as a synthetic lethal hit with ALC1. I don't recall in our studies what our results were. It certainly was not as good of a hit with BRCA as ALC1 was.
Okay. So there's some specificity to ALC1.
Oh, yeah. By far our best hit. Mm-hmm. Yeah.
Okay, the second question is about, do you see differences in olaparib and talazoparib, different trappers?
We do. All right. So, I'll bring up some of these nuances. We do see differences. So, for example, in the BRCA reversion mutants, in those experiments, there really isn't much of a therapeutic window if you use olaparib with ALC1 loss. So when BRCA is intact, loss of olaparib didn't have much. Use of olaparib didn't have much of an effect. On the other hand, with the strong trapper talazoparib, we did. And so what I think is going on is that even when HR is intact, ALC1 loss is creating gap lesions that are creating enhanced trapping of PARP. And in combination with a very strong trapping PARP inhibitor, this can create toxicity even when BRCA is restored.
Mm-hmm. Okay, so the next question is, a very interesting talk when you mentioned about chromatin condensation upon ALC1 loss. I wonder if the condensation involved recruitment of repressive histone modifications as well. If so, can ALC1 be involved in chromatin remodeling in general?
All right, that's a good question. I don't have an answer to it. Parylation is generally a transient type of event on chromatin. All right, and what we observe in our studies with this is that ALC1 is recruited rapidly to damage sites in a PAR-dependent manner. I don't have any evidence that it helps establish a different chromatin state longer term. We also didn't see, at least in the time course of how we did these experiments, didn't see recurrent changes in the chromatin either. But it is still an open question of how is parylation affecting the overall histone changes at damage sites, and would any of them be more persistent? And that's something that we haven't looked into.
Mm-hmm. Okay. The next question is, have you tested cell lines lacking other DNA repair enzymes?
Yeah, so early on, I don't know if the person who asked the question can recall, early on, we did epistasis experiments using the dual Cas9 system. And what we showed is that ALC1 also had a synthetic sick relationship to other genes involved in homologous recombination and increased PARP inhibitor sensitivity. And it did as well with XRCC1, which is involved in single-stranded break repair. All right, but we did not see this relationship with other repair pathways, for example, non-homologous end joining or nucleotide excision repair.
Mm-hmm. Okay. Okay, the last question is, given the minor differences you see with loss of BRCA1 and BRCA2 in the HR pathway, So for example, RAD51 foci, DRGIP assay, do you see differences in the hits in the CRISPR screen with the two genes? Difference between BRCA1 and BRCA2, I guess.
All right. In the library that we used, okay, that's a harder question to answer. So we used multiple different lines, and there were some different hits in each line. AOC1 was common to all of them. And it was a smaller library, right? This is just a chromatin focused library. I think it was 179 genes. So when we have different hits across these lines, it takes a fair amount of work to know, is that specific for BRC1 or 2, or is this related to just other changes in the cell lines? We don't know that. ALC1, on the other hand, was consistent across and was a high confidence hit, whereas many of the others were lower confidence or lesser effects.
Okay, we have a new question. You saw, a couple new questions. Yeah, you saw that part 1, part 2 null cells become resistant to PARP inhibitor, but were those cells very sick too? At least according to, part 1, part 2 knockout mouse study.
Yeah, cells will become sick and will have a slower growth phenotype with combined PARP 1 and 2. You can generate them, and they can be stable in certain settings. So combined PARP 1 and 2 is synthetic lethal in mice. It will reduce viability in cells, and especially in combination with BRC null cell.
Okay. The next question, is there a chance that ALC1 can condense chromatin and inhibit the sliding of replication or causing stalling?
Not that I'm aware of. And I don't want to rule anything out that we haven't looked at. It certainly, in cells, one can observe the decondensation, and that can be observed either by ATAC-seq type of methods, or even in lac operator arrays. The Lerner group showed that if you tether, ALC1, for example, to the lac repressor and fuse it there to these extended lac operator arrays, you can actually visually see this by microscopy. With PARylation, one would predict that it would not be condensing chromatin at replication forks, but I think that remains to be seen.
Okay, so maybe I can ask a second question. So thinking about the mechanism by which ALC1 helped to trap, the lack of ALC1 helped to trap PARP. So do you think it changes the initial amount of PARP being trapped, or is changing the kinetics of release of PARP from lesion?
All right. I think that you're having more lesions there and more trapping. So I think the kinetics would be all right. And then I think that will be the major issue right there, is that there is just more PARP 1 and 2 gets onto DNA.
Mm-hmm.
All right. It is quite interesting of how low you can go with these PARP inhibitors, and that how important this trapping effect actually can be. And if one looks at other factors, then this is not the first example of a gene that of a protein, when it's lost, that increases PARP inhibitor sensitivity, and that it's not through trapping. For example, XRCC1, which is involved in single strand break repair, is not an HR gene, and yet XRCC1 null cells are very sensitive to PARP inhibitors. So if you create these type of persistent lesions like this, it will trap PARP onto DNA and create problems. And if there's enough of it, it can even overcome homologous recombination mechanisms and create toxicity.
Great. Thank you, Roger. I just want to tell you that you're perfectly on time. Right now it's exactly 1 o'clock.
All right. I had my watch.
Thank you so much for your wonderful talk. Yes,
Thanks
congratulations on this work. Yes.
All right. Thank you, Lee. And everyone stay well and stay safe. Thank you.
Thank you.